Welcome students. Myself Arijit Chaudhuri, Assistant Professor, Department of Microbiology. Today we are going to discuss a topic from the SSC syllabus as written over here of semester 4 microbiology honors, that is Aeroicrobiology. Now to start with this particular one, we need to know what aeromicrobiology is. Initially, the term aerobiology was coined by F.C. Mayer in 1930s when he was actually studying the life in air. After that, one part of that aerobiology is termed as aeromicrobiology, where we are absolutely concentrating on microorganisms that are present in the atmosphere. Now, it is the study of microorganisms that are present in the atmosphere, and as you can see, this includes aerosolization, transmission and deposition of microorganisms. And obviously, when we are studying them, we need to study the impact of air microorganisms on the human health as well as the environment. Now, these terms, aerosolization, transmission, deposition, we will be getting all these terms later on. Now, first to understand where we can find microorganisms in air, that is, we do have different stratas of atmosphere above the earth crust, like troposphere, stratosphere, etc., etc. But amongst them, the immediate layer of the atmosphere that is just above the earth crust is known as the troposphere. And this troposphere has got the highest concentration of microorganisms present. It is the most favorable for the survival of microorganisms. Now, when we talk about survival of microorganisms, we mean in this particular strata, their survival is a little better than the other parts of the atmosphere. This troposphere is spanned more or less extended from 0 to 1 kilometers above the above 10 kilometers, I think 1 to 10 kilometers above the earth crust, and this is the region where most of the microorganisms survive. Now, why this is the most favorable region? This is because water vapor is very much present over here, and that aids to the survival of microorganisms because they can prevent desiccation. When you have a little bit of moisture, you can prevent desiccation. And also, if you can prevent desiccation and if you have certain amount of moisture, a little amount of biological activities are also there. So that is why this is the strata of atmosphere where we have highest concentration of microorganisms. Now, when we talk about microorganisms present in atmosphere, one thing we have to remember that there is no native organism in atmosphere. Whatever organism present in the atmosphere are somehow introduced from the lithosphere or hydrosphere of the earth, because in atmosphere there is very little or no nutrients present. As because there is no nutrients, it's not possible to have native organism in the atmosphere. Now they are introduced into atmosphere by some maybe natural activities or maybe by some human activities. Now when we talk about natural activities, it may be a waterfall which can throw some microorganisms into the atmosphere. We can talk about some landslides, we can talk about raining, etc., etc. And we can, if you talk about human activities, then there are many different activities like sweeping, bed making, the running of car on the roads, etc., etc. They are actually throwing or launching some amount of microorganisms in the atmosphere. So that is how atmosphere gets microorganisms. They don't have their native organism. Any native organism is not there in the atmosphere. Okay. Now, why this study is important, or why we are going to study aeromicrobiology? This is important because it has got a huge impact on the human health as well as the environment of this world. Now, why human health and environment, they are affected by the microorganisms present in the atmosphere? Because we do have these organisms, mainly they are the major organisms present in the atmosphere, like bacteria, virus, fungi, dust mites, microalgae. There is another group of algae which is known as macroalgae. It is not part of microorganisms, spores, etc. And they are associated with different animal and plant diseases. Now, as because they are associated with animal and plant diseases, that's why we are mostly concerned about these things, because we humans are having some different kinds of diseases which are coming from these organisms which are present or prevailing in the atmosphere. And plants, they also do have various diseases caused by airborne microorganisms. And as a result, as you can see, that these infectious organisms can cause us diseases and can cause harm to the humankind as well as the plants. So it is very much important for our survival to study these microorganisms in order to understand these activities in a better way. We also have pollen grains of different plants. And these are also very important as because they are present in the atmosphere, and not only that, they can cause severe allergic reactions. In many parts of the world, we used to see that pollen grains are causing severe kinds of human allergies or animal allergies. But we are mostly over here concentrating on the different microorganisms that are present in the atmosphere. Now, when we talk about the diseases, why we are this much concerned about these microorganisms present in the air, the diseases, there are certain of few examples I have provided over here. There are so many different organisms and so many different diseases that can happen from microorganisms of atmosphere, but here are few examples. Now, as you can see, pulmonary tuberculosis, one of the most well-known diseases that is caused by mycobacterium. The names in the brackets are actually showing the causal organism of the disease, and those organisms or pathogenic organisms, they are actually present in the atmosphere. Now, pulmonary tuberculosis, Mycobacterium tuberculosis is the name of the organism. Pneumonia, Klebsiella pneumoniae. Pulmonary anthrax, this is Bacillus anthracis. Whooping cough, this is Bordetella pertussis. Diphtheria, Corynebacterium diphtheri. These are few airborne bacterial pathogens causing certain diseases of humans. Now, some other human diseases caused by airborne fungal pathogens. In air, as we all know that we also have fungi. So, Aspergillosis, Aspergillus fumigatus, and Coccidiomycosis. This is Coccidius immitis. So, these are examples. There are so many different or more diseases are obviously there. Now, human diseases caused by airborne viral pathogens. Now, viral pathogens are very much in news, as you can see that we are suffering from a pandemic caused by a airborne virus, and that is the COVID-19 pandemic that is going on in this world currently. And this particular disease is an airborne disease. It is spread by a virus which is a SARS-CoV-2 or novel coronavirus. That is the COVID-19 disease. This is the newest addition in our disease vocabulary or disease dictionary. But there are other diseases also which are more or same way deleterious or deadly for the humankinds, and those are the diseases. They are also spread through certain viruses which are present in the atmosphere. So, for example, influenza by influenza virus, SARS, as you can say, it is from coronavirus. It's a different coronavirus. COVID-19 is a novel coronavirus, that is the SARS-CoV-2. Hepatitis by hepatitis virus, chickenpox from herpes virus or caused by herpes virus. Common cold, certain picornavirus, dengue fever, flavivirus, etc., etc. There are numerous such examples are there. Over here given a few of them. So, as we can see that there are different diseases that can be caused by microorganisms prevailing in the atmosphere, it is very important for us to understand the whole thing. Now, in case of plants, it's been said that about 70% of the disease of plant diseases are caused by fungi that are present in the atmosphere. So here are a few examples of plant diseases. As you can see, these are caused by certain fungi which are present in the atmosphere, or these are airborne fungal pathogens causing causing disease to plants, like potato late blight, leaf rust, loose smart of wheat, downy mildew, etc., etc., powdery mildew of barley. These are the examples of some diseases caused by fungi present in the atmosphere. Let's move on. Not only these full or whole organisms, sometimes we do have toxins which are airborne, present in the atmosphere, and these toxins, if are ingested by a human being or an animal, it can cause some severe conditions in human beings as well as in some animals. Now, there are certain different types of toxins that are present in atmosphere. Few important examples are given over here. Now, like lipopolysaccharide, this is an LPS, this is called LPS in short. LPS layer, as you all know, that LPS layer is the outermost layer of the gram-negative organisms. And few of these LPS layers can be very toxic in nature, and they are termed as endotoxins. Now, when they are present as part of the microbes or with a whole microbe, that can cause severe damage if ingested. Now, apart from that, these are staphylococcal enterotoxin is another toxin which is sometimes present in the atmosphere and can be very fatal sometimes. And the Clostridium botulinum, another example. This is Clostridium botulinum is producing a toxin which is known as botulinal A toxin. This is also a neurotoxin, and normally this is associated with ingestion of contaminated foods. Now, when you ingest this sort of toxins present in the atmosphere, they can cause acute respiratory symptoms like asthma, coughing, shortness of breath, etc., etc. So obviously, these are another important aspects or another important parts that are associated with microbes, as well as they are very much present in the atmosphere. Now, when they are present in the atmosphere, they are actually suspended in the atmosphere. So microorganisms, when introduced into the atmosphere from some terrestrial or from some aquatic environment, they are present in the atmosphere, they are suspended in the atmosphere, and these suspended conditions or particles suspended in air, they are known as aerosols. So what is aerosols? Aerosols means particles suspended in air. And what are bioaerosols? There are two terms, aerosols and bioaerosols. Bioaerosols, biological airborne contaminants. That means with those suspended particles like dust particles, when you have living organisms, whole organisms or parts of the organisms associated with those aerosols, then you get bioaerosols. We are very much concerned about the bioaerosols as because they actually contain the pathogenic microorganisms present over here. Now, these dust particles or the particles which are suspended or aerosols, they can act as a raft onto which the biological contaminants is riding upon. Now, smaller aerosols, if the aerosol is small, obviously it will travel further in the atmosphere, and as it can travel further in the atmosphere, it can also reach distant locations, and also it can travel further in the respiratory system too. So the smaller aerosols are of greater importance. If larger aerosols are there, which is biological, if added with biological contaminants, that means larger bioaerosols, they are actually suspended a little faster, and they are a little less important than the smaller aerosols that causes harm to the environment and the human and the plants present over there. Now, if we see the aerosols, the Environmental Protection Agency, the EPA, this has divided airborne particulates into two size categories. Large and small, as I told you earlier. Particles which are with diameters less than or equal to 10 micrometer are known as PM10. Particles with diameters less than or equal to 10 micrometer. And PM2.5 are the particles with diameter less than or equal to 2.5 micrometer. So these are the two basic categories of aerosolized particles or aerosols by the Environmental Protection Agency. Now, when we talk about bioaerosols, obviously there should be some classification regarding the bioaerosols. Now, as we have seen that these include the whole entities, bacterial, viral, toxin, etc., etc. These are also classified in different categories. Now, ranges, if the range is from 0.02 to 100 micrometer in diameter, this is the range of bioaerosolized particles. Smaller particles, which are of greater importance, are less than 0.1 micrometer in diameter. So if the particle is less than 0.1 micrometer in diameter, these are known as smaller particles. A little large particle from 0.1 to 2 micrometer are known as accumulation mode, and the larger particles are considered as coarse mode. Now, nuclei mode and accumulation mode, these are the fine particles. These fine particles can cause more harm when they are suspended in the atmosphere along with some biological contaminants than the coarser particles. Now, these particles, we talk about particles, we talk about their presence in the atmosphere. Now, when these particles are present in the atmosphere, as I have told you earlier, that they are launched into the atmosphere. So obviously they are present in the atmosphere by certain mechanisms. Then there is a terminology, that is the transmission. Transmission means their movement from one point to another through the air. And the third thing is deposition. Whenever they are coming in contact with your respiratory system or any other surface, they are getting deposited onto it, right? So, in aeromicrobiological pathway, or what is stated to be the A and B pathway, there are three different phases. One is the launching of bioaerosols to the air, then it's transport, air or diffusion in the atmosphere, and finally their deposition. So launching, transport and deposition are the three events that are actually constituting the aeromicrobiological pathway or A and B pathway. Now let us come to the first one, that is launching. Now launching of bioaerosols may be from terrestrial or from the aquatic substance. Now it can be launched from a particular point. I'm coming to that later on. It can be launched from a particular point, it can be launched from a particular area, etc., etc. So the particular source of the launching can be different, and the nature of the source can also be different. So for example, there may be a point source, say a heap of dust. This can act as a point source wherefrom the air currents are going on and launching some of the microorganisms into the atmosphere. Anyway, it's a continuous process. So that point source would be considered as continuous point source. But when a person, then it's a point source obviously, but that point source is a instantaneous point source. So point source can be of two types: instantaneous point source and continuous point source. Similarly, area sources or linear sources are a little less defined terms. Like if you can talk about a field where cultivation is going on, and in that field during the cultivation process, they are actually throwing or launching some bioaerosolized particles in the atmosphere. The process. Now, these launching is done by a huge area, so it's an area source. If the area is a large area of a dumping ground, it will be continuous area source. Or if it is an area where from, say, the launching is done in some instances, it will be an instantaneous area source. So like that, we can have certain different aspects of launching, how the microorganisms are introduced into the atmosphere. Now let us see this particular figure where we can see that it's a point source. This is a point source, and from a point source, whenever microorganisms are being spread or being launched, they are launched in different directions. Right? So, if a person sneezes, it also forms a cone of spread. The spreading from a point source is mostly a cone-shaped spreading where in different directions the microorganisms are thrown or introduced into the atmosphere. Second point is transport. Transport or dispersion is a process by which aerosolized particles move from one point to another. As I have told you earlier, that this is the transport, the dispersion of those molecules, dispersion of those particles from one point to another, and obviously flow of air, the current of the air is responsible for that. Now, one thing is for very sure that if the particle is lighter, smaller, it will travel more. If the particle is a little heavier, it will travel less. So obviously, the fine particles that we talked about earlier are able to travel more distance than the coarse particles, and that's why they are of greater concern. Now, after this transport process, these biological contaminants can be deposited into a particular area, but when they are transporting from one area to another, depending upon their size and depending upon how much they are moving from one point to another, these transports can be classified into different scales. Sorry, like submicroscale transport. Submicroscale transport is a transport process is under 10 minutes and relatively short distance, under 100 meters. If the particle transports under 100 meters and the time is less than 10 minutes, it will be a submicroscale transport, small transport. Then there is a microscale transport, 100 meter to 1 kilometer distance traveled by the aerosol. Then it's a mesoscale transport, refer to the transport in terms of days. So the particles which are smaller, they can transport for days, and they can go up to a distance of 100 kilometers from the launching portion. So the point where from they are launched, they can even travel up to 100 kilometers. And not only that, there is a transport which is known as macro scale transport or the large scale transport. It can cross oceans. In these macro scale transports, the time and distance are extended even further. That means it can be months and it can travel huge distances like across oceans. They can also transport. Okay. Now, the third terminology, that is deposition, the last step of AMD pathway. That means it has been launched, it's been transported, and now it's going to be deposited. Until unless it's been deposited onto any individual or until unless it has been deposited into any surface, then you are not getting the total impact of this aeromicrobiological pathway. Now, deposition means they are being settled. They are being settled onto any surface, maybe whatever is available in its path. Now, an airborne aerosol will eventually leave the turbulence. Obviously, it will eventually leave the air, ultimately due to certain things, and one of the most important things is the gravitational settling. Because of the gravity, they are finally going down and they are being settled. Now, there is another one, that is downward molecular diffusion. Molecular diffusion is another thing whereby certain molecules are being diffused from one part to another, and that can also lead to the deposition. Surface impaction, if there is any surface onto which the particle collides, then it will be impacted on that particular surface. The surface may be at the bottom, or at the flow, at the path of the flowing, or at the path of the transporting aerosol. Rain and electrostatic deposition. Obviously, the particle is being suspended in the atmosphere. Rain comes. What will happen? The rain droplets will actually take those suspended aerosolized particles into the earth crust. So obviously, rain helps huge in the deposition process, and the electrostatic deposition based on the electrical impulse of the two different things, the particles and the surface on which is being impacted. If they are of opposite electrical charges, then they will attract one another and the particle will get suspended to that surface. So these are basically the processes or the reasons why those transporting particles suspended in the atmosphere are being settled, are being deposited. Okay, so this is the three different steps of the aeromicrobiological pathway. Now let us talk about the survival. They are launched, they are transported, they are being deposited. It may be a few minutes, it may be a few days, it may be a few months. Will they survive the process? Depends. Because in atmosphere, as we have earlier discussed, that there is no nutrients. But in troposphere, we do have a little more moisture, little more relative humidity. Now, if you consider a particle present in the atmosphere under the harsh conditions, like the temperature, like the UV radiation, so it is obvious that they will not survive much in the atmosphere, and as because there is no nutrients for them to grow upon. That's why normally it's a desiccation stress or stress by other environmental factors that actually limits the survival of microorganisms in the atmosphere, right? Now, what are those factors that are influencing the survival? Relative humidity. When very high, that is also detrimental. When very low, that is also detrimental for the microorganisms. Temperature, obviously, temperature is high, then microorganism will survive less. It is to some extent depending upon the capacity of the microorganisms also, that what particular temperature they like to have for the growth and survival. Then radiation, obviously detrimental for them. UV radiation and other kinds of different radiations that are present in the atmosphere, they are also very harmful for the microorganisms. Oxygen. Now, we know that oxygen is beneficial for us, but we do know also that there are different products produced from oxygen. These are toxic, and those products will form due to the presence of oxygen. They are known as the oxygen toxicity. Right? Now, reactive oxygen species are there. So, we are not going to talk about that, but from oxygen, we can have certain reactive oxygen species, and those, when present, they can cause harm to the life, right? So, microbial life is also under the stress because of presence of oxygen. Open air factors. Open air factors means factors that are not absolutely mimicable in the laboratory. In lab, you just cannot produce or reproduce the factors or the combination of factors of atmosphere. So those are also things that are influencing the presence of microorganisms and their survival. But one thing is very sure, that there are certain microorganisms that do have certain spatial ability to survive. They have spatial ability to survive longer in the atmosphere, and these are spore forming bacteria. Bacteria which used to form spore under stress condition. Fungi, they can also produce spore. Protozoa can form cyst. If they are forming these resting structures, then they can survive for a long period of time, because thanks to their specific mechanisms that they have, they can withstand some extreme conditions, and if they can withstand, they can survive much longer in the atmosphere in spite of. So if they can form spores like bacterial spores, fungal spores, or protozoal cyst, they will be surviving more in the atmosphere or a longer period of time than the other microbes who are not having this special ability. And these are the factors that can influence their survival in the atmosphere. Now, we have come to a point where we can right now understand the microorganisms that are present in the atmosphere, what kind of microorganisms they are, what path they follow, the path they follow during the transport process, how come they survive in the atmosphere. Now, there is another terminology, that is extramural aeromicrobiology. Extramural aeromicrobiology is the study of microorganisms associated with outdoor environments. Obviously, when we talk about human health and environment, we do have two different types of environment. Indoor environment that we are currently in, and there is an outdoor environment, right? The outside of the houses or the outdoor environments. Now, the expanse and space present in the outdoor environment are very large. And obviously, in those outdoor environment, the turbulence is also different from that of the indoor environment. Now, in outdoor environment, these expanses, spaces and the turbulence are the two controlling factors of the microbial movement. That means the movement of the bioaerosols. Now, movement of bioaerosols means when a bioaerosol moves from one place to another, if it gets more space, if it gets more turbulent atmosphere, it will go farther. But the concentration of bioaerosols in a particular area will be less in the outdoor environment, as because it's been a flowing or dynamic process, dynamic environment. Now, environmental factors, UV radiations, temperature and relative humidity, as we have stated earlier, these can affect bioaerosols by limiting the amount of time the aerosolized particles or microorganisms will remain viable, obviously, that we have discussed earlier. Now, two most important aspects in this extramural microbiology or extramural aeromicrobiology to be specific, two different aspects are of most importance. One, the spread of agricultural pathogen. Outdoor environment, the agriculture or the cultivation are going on in the outdoor environment. So in those areas, those plants are having or facing the microorganisms that are present in the atmosphere in the outdoor environment. So if agricultural pathogens are being spread, that can be of some importance, and the spread of airborne pathogens associated with waste disposal. Very important thing. Waste disposal is an integral part of our human being, our humankind actually, because regularly we used to dispose huge amount of wastes in the environment, and during the process of this waste disposal, many of the microorganisms, and most of them are pathogenic in nature, they are being thrown or introduced into the atmosphere. So extramural aeromicrobiology is about these two most important things: agricultural pathogens and pathogens associated with the waste disposal. Now the second thing is the intramural aeromicrobiology. Intramural means indoor. In indoor conditions, obviously the turbulence of air will be different, the space is different, and one more thing is different. That is, because of less turbulent air and because of less space, microorganisms can accumulate in indoor conditions. Or if some pathogenic microorganisms are suspended in indoor environment, they have got little chance to get away from that indoor condition. And as a result, if some other individuals, healthy individuals are present in that indoor conditions, they are prone to get that microorganisms to come in contact with them, as because the area is less and turbulence is very less. So limited air circulation, as you can see, and much less radiation. That is another important point. As because in indoor environment, radiation is very less, so that will help in the survival of the microorganism. So microorganisms will be viable or surviving more in the environment rather than in the outdoor environment, and they can be accumulated in a particular area. So the concentration will be higher, and as well as their survival is better in indoor environment. That is harmful for us. Okay. Next, you do have microorganisms, you do have bioaerosols present in the atmosphere. Now, how to get those microorganisms or how to get those bioaerosolized particles sampled? Sampled means if you have to study what are the different types of microorganisms, what are the different types of bacteria, fungi, etc. present, and what is their concentration in a particular area. Given that the sampling is also sometimes very necessary, because we humans have an average height of our respiratory inlet, that is our nose. If that can be assessed, that what are the different concentration of microorganisms and load of microorganisms and types of microorganisms present in that particular area, which are going to affect us the most. That is why sampling is very necessary. Sampling of bioaerosols will give us idea about their identity, not only that, about their concentration and different types of microorganisms that are present in the atmosphere. So this is done for qualitative as well as quantitative assessment. Qualitative means what are the different types, what are the different things present over there, and quantitative, how much they are there, what is the concentration that they are present in the atmosphere, right? Now, common methods. We are not going to the details of these methods, but there are certain common methods that are followed in order to sample these bioaerosolized particles. First one, impaction on solid surface. Now, if you have a solid surface onto which the microorganisms can be impacted, if a known volume of air can be passed through that surface, then after enumeration or after getting the number of colonies or CFUs, you can say you can have an idea regarding the number of microorganisms present in that particular volume of air. So types are also there, and if measured volume can be given, then also quantitative assessments can be done. Impingement in liquid. Air is known volume of air is put through a liquid. So whatever bioaerosolized particles are present, they are getting dissolved, they're getting into the liquid. And when that liquid is tested, obviously at initial stage the liquid will be sterilized, and after the air passes through the liquid, if you are testing it, so whatever microorganisms present in that air, the atmosphere, that will be found in the liquid. So this is a technique known as liquid impingement technique, very important technique and very easy to do technique through which we can have sampling of bioaerosolized particles. Sedimentation or settle plates. By sedimentation process, we can just have the plates given over there, and we can allow the air to settle onto it, and we can have the idea of how much they are present and what are the different things that are present. Electrostatic precipitation, obviously, this is a precipitation whereby electrical charges are used to actually attract the bioaerosolized particles to a particular surface, and after this impaction, they are being measured. This is a flow chart showing how bioaerosols can be sampled. Now, these bioaerosols can be sampled by impingement, collection of particles into liquid medium, as I have told you earlier, filtration, removal of particles by membrane using vacuum. Now, this is another process where you can remove particles by filtration process. What you get is the fresh air, but in the filter you will get the microorganisms. So if you can test the filter, you can get the microorganisms that were present in the atmosphere in the air when you have put into the study. Now from there you can have different techniques used to get the different aspects of your sampling, because if you want to know the identity, if you want to know the amount, so there are different processes by which you can do it. Impaction. Variable particles are collected by a stream based on their aerodynamic diameter. Variable particles can collect in different samplers also. There are different samplers like Anderson sampler, Booker samplers. These samplers use this impaction process, whereby these particles can be collected in the air stream in different strata of that particular sampler, and finally after microscopy or plate count, you can have a proper idea about the bioaerosolized particles. Electrostatic precipitation. Particles are subjected on agate medium by precipitating electric field. Obviously, as I told you earlier, using the electric fields, you can make them impacted on a particular surface, and then you can count them or to see them, check them for their identity and types. Okay, now we come to control of bioaerosols. The bioaerosols that are present in the atmosphere, we know how come they present over there, how come they transport from one place to another, and we know how to sample them. Now, those bioaerosolized particles are harmful for different aspects of human health, for different aspects of public health, for different aspects of laboratory work, for different aspects of operation theaters, etc., etc. And obviously, they are also harmful for agriculture if pathogenic microorganisms are being suspended over there in the fields where the cultivation is going on. For these reasons, control of bioaerosols is a very important thing. How to control them? How we can get rid of these things as much as practicable. We cannot get rid of the bioaerosolized particles fully, but as much as required, how can we do that? Now, there are certain processes or mechanisms which can be used to get rid of bioaerosolized particles to different extents. Firstly, ventilation. The least effective method, it is nothing but to increase the air circulation of a particular place. If you allow air to flow, obviously you are decreasing concentration, right? Like opening a window in a closed environment, use of air conditioners, heat pumps, etc., that can be used in order to increase the flow of air in the particular area. If flow increases, obviously concentration decreases, chance of suspension is little less in a greater flow region. The point is, not only it will decrease the concentration, it may also happen that microorganisms are being introduced with that flow of air when you are opening a window. Now the point is, these are less effective method, but little bit of effective in different conditions. Filtration, unidirectional airflow. This particular process is very much used in the microbiological laboratory in a day-to-day use. We all use this. This airflow filtration is relatively simple and yet a very powerful method to have a particular area which is aseptic or which is free of microorganisms or airborne contaminations. Now, some filters are used like HEPA, high efficiency particulate air filters. These filters are designed that air can pass through it, but microorganisms cannot. There is a specific pore size, and through that pore size air can pass through, but the microorganisms cannot. So when you are having air passed through HEPA, you can be assured that it is not containing any microorganisms. So if it is an indoor or a specific area where you can working under the influence of a HEPA filter, then you may work in an aseptic condition and you can get rid of the unwanted microorganisms to a great extent, right? Now, they are reported to remove virtually all infectious particles, reported to remove or exclude all pathogenic microorganisms or particles, right? Apart from certain very small viruses, more or less every kinds of pathogens are being restricted by the use of HEPA filters. Okay, these types of filters are commonly used in biological safety hoods like the laminar airflow we used to use in our lab, microbiological labs. Again is biocidal agents. That means agents that can kill aerial or atmospheric microorganisms. For example, UV, ultraviolet radiation. Germicidal ultraviolet radiation is an important biocide which can be used to kill the microorganisms present in an air. So if in a particular area which is to be used for a second time after the first use, it can be to some extent it can be very beneficial if the area is lit by germicidal UV radiation for say about 30-40 minutes or 1 hour. So what happens is that microorganisms present in the atmosphere, they are killed by the UV radiation. So these biocidal agents like use of UV radiation may be very much helpful in order to control microorganisms. These are my references for these particular slides. Thank you very much.